Alice just finished eating a big bowl of fruit salad when she began to experience stomach cramps and excess gas. Looking inside of Alice's body, we find the hollow enzyme in a moment of confusion. The hollow enzyme is a subunit of RNA polymers that is needed for the correct attachment along the promoter region. The promoter region is the landing site for RNA polymers that creates a location for transcription to begin. Within the promoter region, there is +1 start site and two essential consensus sequence known as -10 box and -35 region. -35 region and -10 box are consensus sequences that have specific nucleotide sequence that have not changed over evolutionary time, emphasizing that there is an important function they possess. The +1 start site is where the first base is used as a template for the first RNA nucleotide base, marking the beginning of initiation. However, we can see that hollow enzyme is confused because -10 box is missing. The hollow enzyme is unable to align properly with the promoter region due to the absence of the -10 box, preventing transcription from occurring. If Alice did not have the absence of -10 box, the hollow enzyme would not have issues aligning with the promoter region. Sigma factor is needed for RNA polymers to bind to the promoter site by its ability to identify the location of consensus sequences. RNA polymers binds to the promoter region correctly for transcription to occur. Once binding to the promoter, the DNA strand begins to unwind into two strands. The top strand is the coding strand where RNA transcription is not occurring. Coding strand is still important because it can be used to convert into RNA sequence, which can be used to determine proteins that will be produced by the newly formed RNA. Meanwhile, the bottom strand is known as the template strand, acting as a complementary strand for the RNA forming. Before the RNA strand can form further downstream from the +1 site, the sigma factor must leave. Between the DNA and RNA strand, diphosphate bonds are cleaved and a phosphodiester bond is formed, adding the RNA nucleotides onto the transcription site. Without the phosphodiester bond, RNA nucleotide bases would not be attached to the new RNA strand. Still, transcription is terminated by termination hairpin, which creates a stall in RNA polymers, stopping it from continuing to transcribe. Once RNA polymers is stopped, unwinding occurs between RNA and template strand due to their weak bond. Termination hairpin is essential for RNA to continue onto translation and for transcription to cease. Alice had the inability to metabolize sugar, preventing transcription from occurring. However, in real life, it can also cause failure to gain weight, grow, and to even experience malnutrition.